And this is greatly due to advances in DNA sequencing technology. This is a graph of cost per sequencing per megabase pair of DNA over the past decade. A megabase pair of DNA is 1 million bases. In July of 2001, it cost $5,292 to sequence a million base pairs. And just to put perspective on that, the human genome is about 3 billion base pairs. But then something amazing happened in early 2008. Sequencing centers transitioned from the old dideoxy method to second-generation sequencing technology. This decreased the time necessary to sequence the genome and also reduced costs. And by the end of November of 2011, the cost was down to a mere 9 cents per megabase pair. Now, as I said, bacteriophage research has been on the rise ever since, due to the decreased cost of DNA sequencing and the relatively small size of bacteriophage genomes. Medical research into their use as antibacterials has been on the rise, and also at a smaller scale, the Howard Hughes Medical Institute started the National Genomic Research Initiative. It utilized campuses around the country, including JMU, in a class called Viral Discovery and Genomics, or at least at JMU, that's what it's known as. And in the first semester, freshmen and sophomore students take a soil sample from their place of choice, bring it into lab, and they isolate their own novel phage, which was really cool. I did it myself. And then for the second semester, some of the phages from the first semester are selected for sequencing, just a few, and they're selected based on their quality. And for the second semester, the students spend their time analyzing the genome and calling genes and using many different bioinformatics programs. So I started my interest in bacteriophages as a sophomore with Dr. Temple. Now, with all of this influx of new information, we needed a useful way to store it. Famerator, which was written by our own Dr. Steve Kerson, was a bioinformatics program written to do just that. It organizes the genes into families, or groups of closely related sequences. Families is spelled with a PH, not because I can't spell, but because biologists can be funny sometimes. And phage is spelled like a PH, so they wanted to be cute. It is a database of gene relationships, and within the Mycobacteriophage database, there are 305 genomes containing 34,419 genes. So as you can see, that's quite a bit of data. It creates really useful genome maps of the phages based on similarity at the nucleotide and amino acid level. You can see on the diagram, these are 5 different phages. The little teeny colorful boxes are genes within the genomes, and the big color boxes between the phage genomes are areas of similarity between the genomes. The lavender represents an exact match. Different colors towards the right represent lesser matches, and if they're like slightly diagonal, that just means the match is slightly further down in the genome. These families or groups of predicted proteins are based on 2 really important things. First, a 32.5% identity or greater by a clustral W match at the amino acid level, and I'll go into what clustral W is in just a minute. And also, a protein blast expectancy value below 10 to the negative 50, and an expectancy value is also known as an e-value, and that just represents the probability that the match occurred by chance. So the smaller the e-value, the better. There are currently 3,466 families in the database, and it has a mean size of 9.93 members. And 1,348 families only have 1 member. Another bio pun, these are known as orphans. As the number of genomes gets increased, the number of orphans decreases. It was proposed that a microarray be used to do some preliminary genomic comparison and, importantly, gene expression amongst phages. The main steps in a gene expression analysis are first, get the target messenger RNA or mRNA from the specimen, then make complementary DNA or cDNA from the mRNA using reverse transcription PCR. The targets are then fluorescently labeled, oftentimes with dyes known as Cy3 and Cy5, which fluoresce red and green. Then the targets are allowed to hybridize to the probes on the slide. They're added in equal amounts, and the hybridization is governed by the level of nucleic acid similarity. So if the probe sequence is way different than the target sequence, they will not bind, and the color will not be represented. Relative expression or fluorescence is measured for each probe. Now, if you can imagine, a microarray slide is the size of a microscope slide, and there are approximately 10,000 little spots of probes on them. So as you cannot get these results visually, and we have